it's my pleasure to introduce this talk. So this is going to be greenhouse gas measurements in harsh environments and its applications of cavity ring-down spectrometry to uncover ecosystem greenhouse gas dynamics in understudied environments. So with us today is Dr. Christine O'Connell, a postdoc at UC Berkeley in Wendy Silver's lab. Christine is an ecosystem ecologist with a PhD from Minnesota, University of Minnesota, and she's studying greenhouse gas fluxes and emissions in Puerto Rico at the current time. I am the host, Dr. Nickerson from eoSense. I'm the chief scientist and president at eoSense. I did my PhD in isotope biogeochemistry at Dalhousie University here in Canada. And I am one of the co-developers of all of the eoSense products, really. And finally, Dr. Gregor Lucic. Gregor is newly a product manager at Picarro. He did his PhD in volcanic degassing from magmatic volatiles at McGill, also in Canada. And Gregor has 8 years of experience working with Picarro systems. So we're going to go through, during this webinar, we'll be doing three distinct topics. The first is going to be Dr. O'Connell talking about a drought experiment in the Luquillo experimental forest in Puerto Rico. So this is really an analog for climate change and what might be happening to tropical forests in the future. Christine has a lot of really interesting both CO2 flux and methane flux data, as well as some other biogeochemistry that might point to what's going to happen to these types of forests in the future. And she'll take you through that a lot better than I just did. I am going to be discussing some experiments we did here in Nova Scotia using the Picarro GasScouter to monitor greenhouse gas emissions from wetland ecosystems specifically. And look additionally, similar to Christine's experiment, at a local drought that happened here in Nova Scotia. And then lastly, Gregor and I will take you through a little bit of the specifications and the high points of the Picarro and the eoSense systems. We'll follow that up with a Q&A session at the end of this talk. So now I'm going to hand it over to Christine, who will take you through the drought in the rainforest.
Okay. I think I'm controlling the screen. Yeah, I love that thumbs up. Gregor, I'm not sure that I'm like controlling this. Oh, okay, I think that was me, right? Cool. All right, well, thanks for logging on, everybody. Yeah, so as Nick mentioned, my name's Christine O'Connell. I'm a postdoc at the University of California at Berkeley, working with Wendy Silver. So Wendy is a professor at Berkeley, and she has about 20 years experience working in Puerto Rico as kind of the background and context. So my role in this webinar is to kind of provide the sort of scientific case study for using some of these systems in a relatively challenging and quote unquote harsh environment. And specifically, I'm going to do that by walking you guys through kind of a large drought that occurred in Puerto Rico and kind of the biogeochemical responses and the greenhouse gas changes in dynamics in the wake of that drought. So this is all coming from a paper that we published last year. If anyone out there is a tropical forest biogeochemist who wants to look that up. So as background for those of you who don't work in the tropics, there are sort of two big trends that I want to draw your attention to. So the first is that tropical forests house a huge amount of the global carbon stock, and they're really vulnerable to global change. So this is a map of above ground and below ground carbon in biomass. So think like plant biomass. You can see this large red strip across the tropics. So this is a place that holds a lot of carbon. It's also a place with really high turnover rates of carbon because it's warm and wet all year round. So at the same time, tropical forests are also likely to see major shifts in precipitation as climate change progresses. So here you're looking at a map from the last IPCC report that shows the delta, so the change in soil moisture from now, from about the year 2000 to the year 2100. So again, you can see that the tropics have this orange and yellow band, which indicates that there's likely to be a decrease in soil moisture. We know from a lot of other science that that decrease in soil moisture can likely be tied to an increase in frequency of drought, and also critically an increase in the severity of drought. And there are a lot of open questions around drought in the tropics and kind of how that might impact ecosystems. So this study was kind of our attempt in Puerto Rico to take advantage of a huge historic drought and observe what occurred during and after that drought and how we might extrapolate that information forward to know how these tropical forests might shift as kind of the precipitation context for these forests also changes. So here's a graph showing this historic drought that I alluded to. So in 2015, there was a major drought in Puerto Rico. And what you're looking at here is a graph with the date along the x-axis and the anomaly in daily precipitation along the y-axis. So basically everything below the zero line, all of these red bars, is an individual day and how many fewer millimeters of rain that day received in 2015 in comparison to the 10-year average prior to 2015. So as you can see, this drought was both persistent across the entire year. Almost every single day was a negative precipitation anomaly, meaning it was drier than it should have been. And also that it was quite severe. So this is a wet tropical forest that's kind of your standard rainforest, and it can get up to 3 meters of rain per year. So that's a lot of rain. And in 2015, there was only about 45% of the amount of precipitation in comparison to the 10-year average. So imagine a really, really wet environment that's, you know, the plant community has evolved in that context and it's getting half as much rain as usual. So as you can imagine, we were really curious to see kind of what that would mean. So meanwhile, as that drought was occurring and as this ecosystem was kind of emerging from the drought and recovering from the drought, at the same time, we had an already installed field array of sensors that allowed us to take high frequency measurements before, during, and after this 2015 drought. So on the left, you can see a schematic of what sensors we had installed. So kind of the experiment we had previously been doing was really interested in topography. This forest is really mountainous, has a lot of ups and downs, and we wanted to know kind of how topography influenced greenhouse gas emissions. So basically we had overlaid an array of 35 sensor nodes, those are the little orange dots, from a ridge down a slope and into a valley in a patch of this wet tropical forest. So those sensor nodes, each spot had 3 sensors, one for soil moisture, one for soil oxygen, and I'll explain in a reason why that's important, and one for soil temperature. So that's these 35 orange dots. And then overlaid on top of that was kind of a greenhouse gas automated measuring system that allowed us to track greenhouse gases kind of on top of these abiotic sensors that were constantly monitoring the soil. So that's these green circles. So you can see that we had, so each green circle is supposed to be one automated chamber. So we had 3 automated chambers on the ridge, 3 in the slope, and then 3 down in the valley. So those were eoSense automated chambers routed to an eoSense multiplexer, and that multiplexer was routed to a Picarro G2508. So that entire system was going before, during, and after this drought. Now, because we were in a wet tropical forest, this instrumentation had some relatively specific, although not unique, challenges. And this is also something I'd love to talk about in a Q&A with anyone who's curious. So on the left, you're just seeing a picture of our field site. So there were 3 big challenges that we faced in kind of pulling off this sort of system in a mountainous wet forest. So one is that a tropical forest is just a super hectic environment. It's really dense, trails get overgrown easily. You're hiking this equipment up a mountain basically, and it's very kind of challenging to physically move around. The second was that we were in a remote field location, so there was no line power. And instead, you can see this lumpy red thing in the left-hand photo is a household generator that we used to power this entire system for several years. So you can see we have our little Home Depot shed with a little tarp in the middle of the rainforest with a generator and some camping chairs. So that was kind of our top of the line setup. And then the third big challenge is this is a wet rainforest, and a lot of this instrumentation doesn't always like being around a lot of water. So we had a lot of challenges around flooding issues, especially in these valleys, and kind of air moisture and humidity issues as well. So a lot of these challenges will definitely relate to those of you who aren't working in the tropics, but who are working in, for instance, wetlands, flooded delta environments, or any other really remote mountainous system that doesn't have line power. Again, something I'd love to talk about in the Q&A. Okay, so on to the results. So the first big result from our drought was that there were kind of immediate and threshold consequences to important abiotic factors. So this time series graph has on the x-axis late 2014 to early 2016, so across this 2015 year of drought and a few months on either side. And then the y-axis, you're looking at soil moisture. The different colors indicate where on this ridge to valley kind of topographic transect you were. So warm colors are valleys, cool colors are ridges. And these dotted lines indicate the onset of drought and kind of the emergence from drought. So this first dotted line shows kind of when the onset of drought occurred. And you can see that there's an immediate stark threshold effect in which soil moisture dropped precipitously. Conversely, there was a really long recovery for soil moisture to kind of revert back to even close to its original levels. And in fact, this recovery was so long that statistically, we found that there was kind of a fourth period of drought that was statistically different than the other periods. So these dotted lines kind of break up the different statistical periods of drought that we found. So the first is pre-drought, so kind of our baseline. This second period is during drought, so we're calling that the drought period. Then this third period, which we were not expecting to find, is a kind of statistically distinct drought recovery period. So what's interesting about that is this is during the period in which rain had started to return. So from an ecologist point of view, if you're just measuring rainfall and you think a drought is over, one thing we learned is that kind of below ground doesn't care. Below ground thinks that this drought is still happening. And kind of as far as microbes and roots are concerned, you're still in it. And then finally, we did find this fourth period that was kind of a distinct post-drought era. So when it comes to soil oxygen, we saw a really similar effect. So this stark threshold effect in which soil oxygen increased. So that occurs because when soil moisture goes down, oxygen is able to diffuse into soil pore space much more quickly than when there's water there. So when soils get drier, they tend to get more oxygenated. And you can see during drought and drought recovery, you had this really long recovery time. And in fact, for oxygen during the post-drought period, other than in valleys, you never saw these soils recover to their previously oxidized levels. They stayed oxygenated, kind of even though moisture had reverted roughly back to baseline. And I mentioned before that I would explain a little bit about why we care so much about oxygen. So we know from a lot of previous work that in the tropics, redox reactions, so reduction oxidation reactions, can have really strong controls over biogeochemistry. And in this forest specifically, iron is a really big part of that. Okay, so we were following soil moisture and soil oxygen, but we wanted to know more about kind of what was happening to nutrients. So what you're looking at here is a graph showing that inorganic phosphorus, so the kind of phosphorus that's both limiting in the system and also readily available to plants, saw major declines after drought. So on ridge, slope, and valley, in all three of these topographic zones, from pre-drought in orange to post-drought in red, there were statistically significant declines in inorganic phosphorus. At the same time, there were statistically significant increases in organic phosphorus, and we attribute that mostly to changes in decomposition rate. But organic phosphorus isn't readily available to plants, so even though there's still phosphorus in the system, it's not the phosphorus that can help with plant regrowth and kind of plant community recovery from a drought. At the same time, iron 2, the reduced form of iron, saw big declines after drought, and that makes sense because it's not as wet anymore. At the same time, iron 3, the oxidized form of iron, saw big increases after drought, especially in valleys. Now we care about that because iron 3 is relatively sticky to inorganic phosphorus. So what might have been happening here is that inorganic phosphorus, at least in valleys, was being bound up by the newly oxidized iron. All right, so on to the greenhouse gas results, which I know everyone's here for. Okay, so we followed carbon dioxide and methane from before to after drought. So what we're looking at here is a carbon dioxide graph. So on the x-axis, we have our drought time period, pre-drought, drought, drought recovery, and post-drought, which I'm hiding for a second. So one of our big trends that we observed is that during drought and drought recovery, slope, these purple bars, saw a huge increase in CO2 emissions. Valley also saw a large increase in CO2 emissions during drought. Now that makes sense. Those are the wetter parts of this system. So during drought, when there's less soil moisture, any kind of anaerobic control on that's kind of keeping aerobic respiration down is relieved. So that was one of our big trends, this big increase in CO2 emission from slopes and valleys during drought. Ridges behaved really differently. After drought is when they saw this big increase in CO2 emissions. But in general, kind of across the board, drought led to big increases in how much carbon dioxide was coming out of this landscape. Now, methane, we saw a really different pattern. So again, pre-drought, drought, drought recovery, and post-drought. So pre-drought, kind of everything's behaving as we would expect it to in a tropical forest. So valleys are huge emitters of methane, because they're wet, they're anaerobic, and they're also warm, and they have tons of carbon in them. So lots of methane coming off of these anaerobic wet valleys. At the same time, ridges, which are relatively oxidized or relatively oxygenated, are taking up a slight amount of methane, so they're net consumers of methane, and slopes are somewhere in the middle, they're about neutral. Now, during drought, we saw two big trends. One is that valleys, the amount of methane they were emitting just plummeted, although they were still net emitters. But at the same time, the ridge and slope sinks got a lot larger. So during drought, we're seeing all of this methane, which is a really powerful greenhouse gas, being taken up by most of the landscape, slopes and ridges, and valleys not emitting very much at all. So we did a calculation and kind of normalized everything to CO2 equivalents, and this shift in methane was enormous on the scale of this entire drought. So this is a big kind of carbon savings to drought. So that would have been a great story. And if we hadn't kept measuring, probably our headline would have been, drought is good for greenhouse gases. But in fact, because we had these high frequency measurements, we were able to keep measuring methane, and we saw this huge rebound in methane emissions after kind of this post-drought recovery period. So valleys immediately rebounded to their methane emissions from before drought. That makes sense. What we weren't expecting is ridges and slopes to become huge net emitters. And in fact, over only a couple of months of post-drought period, 99% of the carbon savings from all of this uptake of methane on ridges and slopes was completely washed away. So the methane story went from being, okay, drought leads to this big methane sink, to, as far as methane is concerned, drought is a wash. So a quick point I want to make about the measurement context. As you can see, other than pre-drought valley, basically all of these error bars are really tight. And I spent my entire PhD working primarily with closed chamber measurements, and getting this kind of accuracy was basically impossible because methane is really flashy, and it just, the amount of kind of human labor that would have had to go into that would have been basically impossible to do. So in this study, if we hadn't had these high frequency greenhouse gas measurements, I honestly don't think that we would have been able to accurately assess this temporal trend, just because we probably wouldn't have had a large enough sample size. So these sorts of rapidly changing natural global change experiments seem like a place where they're a really good fit to kind of deploy these sort of data-rich instrumentation systems, because things can change quickly and in ways you're not expecting. These sorts of natural experiments are new, unfortunately, / fortunately for scientists. And kind of capturing all the dynamics is really hard to do if a priori you're trying to figure out what your experimental design is and you only have, you know, so much graduate student time or technician time. Okay, so our two big takeaways were that drought is very likely to impact tropical forest carbon cycling via two routes: directly impacting tropical forest carbon via these changes in greenhouse gas dynamics, and indirectly via a decrease in inorganic phosphorus availability, with kind of these knock-on effects to future tropical forest storage as, you know, net primary production kind of kicks back up after drought. So yeah, one of our big takeaways is that drought and rainforest probably has really complex direct and indirect reactions that of course are also interacting with each other. And finally, the last note I'd like to leave you on is just that following these natural experiments is going to be really critical, especially in tropical forests, where we both know very little about how they're going to be impacted by global change, and also where they are kind of really important stores and stocks of carbon and really important controls on global biogeochemical cycling. And with that, I'd like to say a few thanks. The Silver Lab has been fantastic, and this was a really labor-intensive project, and we had a lot of technician help, and we could not have done it without them. So thank you to all of our technicians. And with that, I think I'm handing it back to Nick, and then I'm happy to take questions at the end of the hour.
Okay, thanks, Christine. Gregor, have you reshared? Yes, excellent. Okay, perfect. Okay, so, you know, Christine kind of stole my thunder on a lot of the points that I'm about to make, but that's good. So what I want to talk about now is a little bit of follow-up from that experiment that Christine did, and sort of some of the learnings we got from that, specifically around how to deploy in you know, tricky environments. And to talk about that, I kind of want to focus on wetlands as being another environment that's difficult to get, you know, good measurements in, and it's difficult to deploy in because you generally don't have access to line power. You have a very, very wet, sort of harsh environment that you're trying to get into. And, you know, the other thing I think is really interesting about wetlands, and so here are some figures by in a paper by Turetsky et al. From 2014. We have a, you know, a pretty good global database about what wetland methane emissions look like. But, you know, as with everything, it could stand to be improved to an extent. But the interesting thing about methane emissions is for me at least, that they can go basically from methane uptake, like Christine showed, all the way to, you know, massive methane emissions when you start to get ebullition events. And so you can see from this table that I've got up here that, you know, the mean methane fluxes, we have a good constraint on what those look like. You know, you can look at the error bounds here of, you know, plus or minus 6.2 milligrams per meter squared per day. That's pretty good. But what we don't know a lot about is what the distribution over the course of the day or the course of a year in those methane emissions looks like. So you can see, if you look at the maximum methane flux over here on the right, that we tend to have a very long-tailed distribution where we get some very large fluxes compared to what the mean or the median fluxes look like. So I think both because of the you know, the ability to deploy these systems in harsh environments like wetlands, and because of the continuous data that you get with these types of systems, we could get a lot more concise answers on what methane emissions look like globally. And like Christine said, they are very sporadic. So it's possible that this data, you know, we may be missing a lot of these high frequency events because of the things like grad student labor and people generally not going out into the field during the rain and so on. So I just wanted to say that, so this is Christine's talk was using the G2508, which is the 5-species Picarro analyzer. Mine is going to be mostly focused around the GasScouter. And if after this webinar you want to know more, we have several blog posts up on the eoSense website, so www.eosense.com. going through most of what I'm going to be presenting today. So the reason we went, away from the G2508 and to the GasScouter for these experiments is because, wetlands are they're difficult to access, especially if you don't have a nice, boardwalked wetland. So the GasScouter is nice and light. It measures CO2 and methane. But I think that the most important factor here is the power consumption of the unit. So we've got a 25-watt power consumption, which means, unlike Christine's experiment, we can start to think about running this system on some batteries and a solar panel array that's fairly modest. Like, we're not dragging 20 or 30 solar panels into the field. This is 1 or 2. And then the other thing that's great about the GasScouter is that both the resolution on the CO2, which is 0.4 ppm, and the resolution on the methane, so 3 ppb, and the measurement range, so 0 to 3,000 parts per million and 0 to 800 parts per million, make it really suitable for these wetland environments. So you're never going to top out your chamber measurement, and you get enough resolution to resolve even the smallest of the fluxes that you're going to see. So this is going to be a little bit different than Christine's. It's going to be more of how we went about setting this up and then getting into the data. So, the GasScouter, like I said, is 25 watts of power. So we went about looking at our latitude here in Nova Scotia, which is where we did the experiment, right where my mouse is hopefully pointing. So in the summer, we get about between 4 to 6 kilowatt hours per meter square per day, which means we would yield somewhere in the ballpark of 136 to 204 watts of solar power per day. And in the winter, you can see it goes down a bit, but it's still a modest solar panel size. So I mean, so what I'm talking about here is the total amount of watts of solar energy that we need to keep this system running per day. So somewhere, depending on winter or summer, between 136 to 408 watts, which is, if you're getting 100-watt solar panels, it's between 1 and 4 solar panels that you need to take out to the field to deploy. And for these experiments, we, batteries are heavy, so we, more or less came to a conclusion that 3 days of autonomy, so 3 days with no solar input whatsoever, would be kind of a suitable trade-off for this experiment. So that's about 2,450 watt hours of battery power, which, if you're using a standard 12-volt battery, is about 200 amp hours. So 2 deep cycle batteries and 2 solar panels is what we decided was, a reasonable system to get this thing into the field, into a wetland environment, and have it running pretty consistently. So what we did is we went about making another Home Depot style shed for this system, just to keep the direct rain off of the GasScouter and the eoSense gear. So we just went out to the local hardware store. We picked up what I believe is a garden shed to store tools. We cut a couple holes in the side of it to let the tubing out and let the connections to the solar panel out. We wired it up with a pretty standard charge controller just to regulate the voltages coming into the battery. And you can see here we've also added 2 wires. So 1 wire to power the GasScouter, 1 wire to power the eoSense multiplexer in the system. And then, so in order to do this shows 3 batteries. We only use 2, but we basically just hook the batteries up in parallel. So we get our 12-volt supply, and we have a lot of amp hours to draw on. And in the end, this is what the entire system looks like. So compared to Christine's system in the rainforest, which was relatively complex, this, I think once I got it out in the field, it took me maybe 30 minutes to get set up. And there's, I'll show you one issue with it in the future, but it was relatively straightforward and kept the system running for the most part for the whole field trial that we were doing. And yeah, in addition, it kept the rain and everything off the GasScouter. So the experiment itself was set up in an experimental wetland just north of us here in Truro, Nova Scotia. So you can see here on the right-hand picture, there's a large bridge running through different cells of the experimental wetland. And in the different cells, they're just treating different types of water to determine what the biology can do to the composition. So we set one of the eoSense chambers up on the ridge here to just measure we expected, mainly CO2 emissions from the ridge because it's a nice dry and a very steep slope. And then down here in the wetland area, there are 3 chambers nestled in among these in reeds that we were using to look at, more closely CO2 and methane flux from the wetland environment. And so here, here's an example of the data we got back. So this is when I alluded earlier to the more or less continuously running. We did have a period of time where, maybe for the first time, we discovered that there's a fuse in the Picarro GasScouter backpack that can blow, and we, subsequent to this, we put a protective fuse in front of the system to make sure that this didn't happen again. But what we see in the data is not that surprising for the CO2, but more surprising for the methane. So here in the red line, we have temperature throughout the day. The black dots are the chamber that was on the slope. So the one that we should have been expecting to have CO2, but maybe no methane. And green, blue, and red are all the 3 chambers that are within the wetland environment. So what we see is that all of the chambers are responsive to temperature for CO2 flux, which is what we would expect. We see that the interesting thing about the CO2 data is that the behavior, so sort of the Q10 behavior, I guess, is different between the ridge and the wetland environment, where you have a higher response to temperature in the wetland, and I would imagine it's likely due to moisture availability in that wetland environment, whereas the ridge was quite dry and probably moisture limited during the course of this experiment. But the CO2 data, generally, other than noting that, we do have a lot of statistical power, even from the data that we've managed to collect in this experiment, we're able to see statistically significant differences between the ridge and the wetland fluxes. Maybe the more interesting thing is, and I think this is maybe why wetland scientists would want to do this, is the methane emissions. So you can see in this figure, once again, black is on the ridge, and black is actually on the right-hand axis here. So it's CH4 uptake consistently. Whereas blue, green, and red are all within the wetland environment. So what we see is the ridge, not surprisingly, has a small but consistent methane uptake, according to the GasScouter data. And the wetland environment has a decently sized, I think it's in fact maybe about the same size as Christine was showing us, of CH4 emission. And what I really want to focus on is, so there is a bit of what appears to be diurnal variability in one, maybe 2 of the methane emissions, likely related to temperature. But for the most part, during this first period, the methane emissions are fairly flat, fairly consistent. What gets interesting, and I guess the other thing I should mention is that this is on a log scale. So note that. What gets interesting is over here. So here we had on the right-hand side of this plot, we had a rainfall, or several rainfall events, in fact. And what we see is some pretty massive spikes and, in fact, kind of troughs in methane emissions during that rainfall event. And, I'll show you in the subsequent slide, but what we expect is that's due to a combination of pressure, so increased water pressure, pushing methane into the pore space in the wetland, and then potentially some volume replacement of air, kind of causing ebullition from those wetland chambers. So this is why we think that. So On the left-hand side, so this is a single chamber measurement from that same experiment, and it would be from the end where it started to rain. And on the left-hand side, you have a CO2 flux measurement. So this is the concentration time series from the eoSense software. And you can see that's a really nice, clean, linear or exponential fit to that data to yield a CO2 flux. And on the right-hand side, from the same measurement, is the methane flux from that site. So going back to what Christine said, if I was using a GC, for example, and I had 3 or 4 measurements, I would probably throw this data away personally. Because I look at it and I think, okay, there's an increase, but it's kind of all over the place. But what's really interesting about using the GasScouter data is that we're able to see, hey, okay, you know what, the CO2 data is clean, which means we know that it's not an issue with the chamber or the measurement itself. The methane data, what it looks like, and you can see it at least twice in this curve, is that we have an ebullition event right at the beginning of the time series. So right here is the first ebullition event. You have a small increase after that event. And then here you have the second ebullition event, and then maybe more of a diffusive methane flux after that. So, the power of having that really finely time resolved data is to be able to pick out things like this, things that would fall on that right-hand side of the histogram of wetland emissions, and to start to think about those events more more scientifically, I guess. I mean, I think that right now, especially with static chamber measurements, people might be wary of data that look like this, but with the full suite of measurements from the continuous instruments, we were able to actually start thinking about these more critically. And so the second study we did with the GasScouter was to, instead of having it running continuously in the field, what we wanted to do is kind of evaluate the potential to take it out and do a survey style measurement. So once again, this is up on the eoSense blog if you want to know more details about it. But we basically did a little bit of finagling with the wiring inside the GasScouter and turned it around so that the power from the GasScouter was coming out to one of our eoSense eosAC chambers and was able to run it in the field with a little bit of software on the other side run by the cell phone. So there's a picture of me next to the bog, and so this is kind of how this comes, maybe links back to Christine's experiment, was that this was also during a drought period in Nova Scotia, which we don't get very often. But this is a bog out near the coast. It's usually very saturated with water. And yeah, I don't think my feet got wet once during this, but we kind of wanted to look at how the different parts of the bog, which is this next slide, responded to that, decreased moisture availability. So we selected 5 sites across this, the bog. So we've got a pond site over here, which is right next to a water body that's, it's not where it normally is, but it still has quite a bit of water in it. We've got a second site in a spruce tree sort of environment. We've got an upland site, which is, so this whole area is underlain by granite. So the upland site is mostly rock with a few really tough species hanging on. And then we have a bog environment down here, bog bank. So this is right on the fringe of the bog, and then within the dried portion of that boggy environment. And so what we saw from this experiment was maybe not surprising, but what we like about it is the precision with which we were able to see this. So we saw a reasonably high CO2 flux on the pond bank, in the uplands, and within that spruce tree area, and kind of what you would expect, which is that the highest fluxes came from the areas that had those spruce trees that were probably emitting a combination of microbial emissions and root emissions of CO2. And then as you get into the bog bank and the dried bog, the CO2 fluxes tend to go down. Whereas, near water bodies, so the pond bank, the bog bank, and the dry bog, we tend to see higher methane fluxes from this side. But the critical part of this plot, I think, is not the fluxes themselves, but rather the precision with which we were able to resolve these. So this is only 2 measurements taken using each chamber, or each location, sorry, only has 2 chamber measurements, each about 7 minutes long. And the error bars, which I don't have on here, but they're less than 6% error between the 2 measurements. So you get very precise and very repeatable measurements from this system. I think the only time we saw a non-repeatable measurement was when we stepped right around the chamber trying to see what we could get to come up, basically. And so just to contrast that with what we saw in the previous study, so here's an example of data from one of those sites, I can't remember which one, showing very clearly a very nice, try not to get too emotional about it, but a very nice increase in CO2 emissions, or increase in CO2 concentration in the chamber over time that fits quite well to an exponential function. And then this, this is maybe the more, this is the newer part of it, which is a very, very nice increase in methane concentration within the chamber head space over time. And so, I mean, you probably wouldn't expect to be resolving things on this level from a static chamber measurement using GC or vials. And not only that, but with the number of data points we get. So I think the GasScouter, and Gregor can maybe correct me on this, but I think it's reporting data every 1 to 2 seconds. So we have a lot of data in here to add statistical power to those fits and really solidify what those fluxes look like. And so after that, we, as part of the follow-up to that experiment, we kind of went to the drawing board a little bit and looked at a paper by Christensen et al., which I believe is in agriculture and forest meteorology. But we calculated the minimum detectable flux for this system. So the combination of the GasScouter, the auto chamber, and the eoSense multiflexor. So those are really there just for the volume component and the surface area component of that calculation. But what we find is, we have very good ability to pick up CO2 emissions. I mean, what we're talking about here in the table is, with a 1 minute measurement, you can resolve a flux as low as 37 nanomoles per meter squared per second. And similarly for methane fluxes, with a relatively short measurement, which this, this is corroborated with our data, we're able to get, 0.28 with a 1 minute measurement, or 0.06 rather, with a 5 minute measurement, nanomoles per meter squared per second. So we're able to resolve, fluxes that I would be very confident in saying that people would have reported as a zero flux before this kind of technology existed. And so the other, relevant point is the GasScouter is unique in this, in that it was, maybe the MDF limits when we calculated them for other instruments were, maybe double the GasScouter, up to 100 times larger than the GasScouter's minimum detectable flux. And so this is all based on theory, obviously, but I mean, like I said, the data and the theory corroborate each other based on that experiment we did at Poly Cove and these calculations. So I just wanted to come back at kind of the end to this slide, which is, again, like I said at the beginning, I think that these instruments have a lot of promise for remote, harsh environments. And in particular, both Puerto Rico shows this, and also the environments we're looking at here, environments where fluxes are sporadic or are, they're non-trivial to interpret using older techniques, like this ebullition event that we see on the right. I think we have a lot of opportunity to really get more robust data sets with these types of instruments, and start asking some trickier scientific questions if we have the data to support them. And so just before I hand it back over to Gregor, we're just going to go into the more technical specifications portion of the talk. So just an introduction to the automated chamber. So this is the same chamber that was used in Christine's study, as well as the two studies I presented. So it was really designed to measure multi-species gas flux. We designed it as well specifically around integrating with the Picarro analyzers down to the extent that the multiplexer is in fact the same footprint as the analyzer, so it'll sit right on top. We can connect up to 12 chambers to these multiplexers in a closed loop recirculating mode. And the other thing is that we also have a 30-meter reach from the analyzer, which I think is better than a lot of other systems. So you can really spread out your field site and get some good data from that. You know, the other thing that we found that is great about the chamber is that it's got a small footprint and a vertical lift system, which lots of people like because it doesn't get tangled up in vegetation, and they can put it between rows of cropping systems and that sort of thing. And we also offer so in addition to the multiplexer control software, we offer the eosAC AC software that does all of the flux calculations. And if you have an isotopic analyzer, it will do killing plot analysis for you. And finally, we have fairly easy integration on the back of the auto chambers with other peripheral measurements. So we can throw in soil temperature, soil moisture, and kind of pull all that data together, all of it stored on the Picarro instrument. And ready to go when you hit the process button. And so now back to Gregor.
All right. Thank you, Nick, and thank you, Christine, for the lovely presentations. I want to give you guys just a very quick overview of the products that you saw today, a little bit about the technology and some of the other things that we can do with them. So for those of you that are not familiar with us, Picarro, we make laser absorption spectrometers that work based on cavity ring down spectroscopy. So if you want to learn more about the technology, sort of the nitty gritty details, I invite you to visit our website. And the technology that you saw today utilizes sort of a near infrared laser platform, which ultimately leads to good precision, which you've heard about today, but also long-term stability. Both types of systems are portable. They have been deployed on boats, on airplanes, in cars, carts, and sometimes even animals. And again, if you're wondering how that exactly worked, I invite you to look at some of our sort of testimonials and customer pictures on our websites. Some other advantages that are good about these systems is that they have low calibration requirements. So standards are readily available. They have relatively limited maintenance and no real consumables that they require for their operation. And as part of sort of today's seminar series or the webinar, you saw that they are compatible with a suite of peripherals, but we were highlighting chambers in particular. I wanna just briefly highlight two of the products that you saw today, and then see how they fit into our overall scheme of products. So we started off with Christine's presentation where you saw the G2508. So this is a multi-species or a 5-species analyzer that can measure CO2, methane, N2O, ammonia, and water at the same time. And we also have the option of adding an O2 sensor to it. So you can measure varying oxygen compositions inside the chambers. It has very good long-term stability and precision for all 5 species. And in a lot of instances, when you look at laser spectrometers, there is a lot of what we call cross-species interference. So if you have variable CO2, variable methane, variable water, and the scientists here at Picarro in-house have done a really good job at applying these corrections for you, so that if any of these species vary daily, monthly, the system is automatically correcting for any effects on it. And ultimately, I think what you've seen is that the systems are compatible with other peripherals. You can connect external sensors, and you can also wire these things into data loggers if you already have any existing infrastructure. On the other hand, Nick presented more on the GasScouter, which is based on the same technology, but certain modifications were made to make it more portable, transportable. So it can measure CO2 and methane and water at the same time with some of the best-in-class precision for those species. We also have a methane-ethane model available for those that are interested. It's got good portability, good battery life. It's up and running in under 5 minutes. And you can attach a single chamber, sort of a survey type thing where you walk around and place a chamber on the ground, or you can place them in sheds like Nick's, or even potentially Christine's in the future. But ultimately, I think one of the takeaway things that I want you to have from this talk is that the Picarro products are quite modular, so you can pair different things to them in different arrangements. And I wanted to highlight just 3 examples for you of how or what you could potentially do with them. So on the one end, we have sort of what we're calling real-time multi-species. This is where you can have an instrument like a G2508 in a more stationary setting. You can have short-term or long-term automatic chamber deployments. And the good thing is that you're not restricted to the G2508. You can have you can measure other species, you could measure even isotopic changes with time. So you can mix and match as much as you want. And again, single chamber, multiple chambers, it's all compatible. And you really want to use these systems if you're looking for sort of high precision, really low detectable fluxes, if you want to measure multiple species at the same time, and you can pair all sorts of chambers, even your own, to them. In the middle end, sort of, we have the mobile solution. So this is something you would use the GasScouter for. You can use it with static chambers, dynamic chambers. You can walk around with them, and you can get the dual species from them. So again, even though it is portable, it still has relatively good precision, good low detectable flux, dual species, and so on. And while sort of the majority of the talk today was spent on sort of these measurements using dynamic chambers, we sort of realized that in certain cases, it's also sort of, if the field requirements are more restrictive, we have the option to measure discrete samples as well. This is for both concentration and isotopic measurements. And I invite you, if you're interested more about the performance of these systems, the applications, to visit our website and get in touch with us. We have a lot of materials available that will help you if you're familiar in this field, if you're new to this field. And we have people that are here at Picarro that will be more than happy to chat with you about your application. And thank you for visiting sort of the webinar on behalf of Picarro. And I'll hand it back over to Nick for the very last section.
Thanks, Gregor. So I think we're just going straight into the summary here. So you know, you've heard a lot about the systems over the course of this webinar. So what we're saying, you know, high precision analyzers with trace gas capabilities, and particularly these low power analyzers, have opened up a lot of new doors for monitoring in remote and harsh environments. You know, as Christine said, and I think as I showed in mine, you know, coupling these with an automated chamber system can provide you with really powerful greenhouse gas emissions data sets. that really, you get the statistical power to understand and monitor difficult or never seen before, in some cases, processes happening, biogeochemical processes. And the last point I want to leave you on is that improvements, and I'm sure that there will be more improvements in power consumption, portability, and the kind of the robustness of the systems, the ability to take them to the field without worrying about, maybe dropping them once or twice, is going to allow for, more and more greenhouse gas measurements to be made in remote locations with, comparatively minimal infrastructure requirements. So you think back to that map I showed at the beginning of where we actually have, methane flux data for wetlands. I'm hoping to see that these systems will greatly improve that map over the next few years. And with that, I think we're straight into the question period. So thank you all for joining the webinar today.